Hello everyone and welcome back to Biology with Mrs. Evans. Today we are going to be looking at enzymes in this video. So we already talked about enzymes briefly when we talked about proteins. So some of this is going to be a quick review because we already talked about this. Enzymes, remember, are a protein, and proteins, remember, are made up of amino acids, and enzymes are what we call a catalyst, a biological catalyst, because they speed up chemical reactions, and they do that because, remember, they lower activation energy. So I have the graph here. We already saw this graph once before. So in blue, this would be the normal uncatalyzed reaction, and you can see that it would require basically this much activation energy. As soon as you add a catalyst or an enzyme, notice that the activation energy decreases significantly. So again, enzymes speed up chemical reactions by lowering activation energy. Remember when we talked about them, I said that there were two types. One is anabolic. Anabolic enzymes build larger molecules. An example of that is DNA polymerase. And the other type is a catabolic enzyme, and catabolic enzymes break down larger molecules into smaller molecules. And I said that the way that I remember that is because of cats being destructive, like to break things down or break things up, rather. So catabolic enzymes break larger molecules into smaller pieces, and then anabolic, just think of an anvil, they take smaller pieces and build larger molecules. So I have this diagram on the screen because it basically shows you how an enzyme works. So here's your enzyme, and notice that it has this little space right here. This is called an active site, and it's based on shape as to how it functions. So only something that fits into that specifically is going to basically work, or the enzyme is going to work on, and that's called a substrate. So notice how for this particular one, this is a anabolic enzyme, because you see you have two smaller pieces coming in, two substrates, and what happens is it's going to bind right here to the active site, and then the enzyme is going to make or do its magic. So what it's going to do is it's going to form a bond between substrate 1 and substrate 2 to produce this new product. So we're going to go into a little bit more detail about that in a second. So again, another picture representation of how an enzyme works. So usually what we talk about is an enzyme is waiting for a substrate to come along that is going to fit into its active site. And enzymes are specific, meaning that it's only going to work on certain substrates because they fit the active site. So notice this enzyme is waiting for these two specific substrates to come along because they fit right here in the active site. And then once they basically bind together, the substrate binds to the enzyme, we call that the enzyme substrate complex that you see here. Usually we don't refer to it as an enzyme product complex, because we just refer to it as enzyme substrate because it's going to do its magic. And then what happens is the product is going to be released, and then the enzyme is free for another substrate to come along. And remember, I talked about this in class, that think of an enzyme as a bus, a school bus. The school bus is going to pick you up, take you to school. When it drops you off, it then can go and pick up other students if it's doing a second run. It doesn't magically turn into a car or a motorcycle. It stays a bus. So the enzyme is going to be left unchanged by the reaction that it helps basically take place. So enzyme action, you need to be able to explain how an enzyme works. So the first step is you have an enzyme that is basically waiting for a substrate to come along that fits its active site. Once one comes along, it is going to bind to that substrate at the active site. So here's the picture representation. You can see the substrate comes along, and it fits right here in the active site, and then the enzyme is going to do its magic. If it's a catabolic enzyme, it's going to break bonds to create basically two new products, and if it's an anabolic enzyme, it's going to create new bonds to create a much larger molecule. So once it binds, then it's going to do its magic. Like I said, it's either going to break bonds or it's going to form new bonds. So it's going to basically convert the substrate into the new products. And then the last step is for the enzyme to release the products. So the enzyme has done its job. It's either formed bonds, broke bonds, depending upon what type of enzyme it is. It releases the products, and it's waiting for another substrate to come along. Now, because of the way that enzymes work, you'll hear it referred to as what they call a lock and key method. And that's because you can't use your car key to open up your house key and vice versa, because they are designed to fit specific locks. And that's exactly how enzymes work. They only work on specific substrates because they fit their active site. Now, enzymes are dependent upon their shape. So if there's anything that alters their shape, it's going to alter their function, which we're going to talk about in a second. So let's review. You have the picture here. Notice you have a free enzyme that is just sitting there waiting for a little substrate to come along. This substrate is free. Notice it fits directly into the active site. So the substrate comes, bonds to the enzyme at the active site where it fits, basically. The substrate is going to bind to the enzyme, creating the enzyme substrate complex, which is this. And the enzyme is going to do its magic. It's either going to create new bonds or break bonds to create the new product. And you'll notice this one, because of how the product is released, this one is a catabolic enzyme. So it took a larger molecule and it broke the bonds here to create the two products. And again, once it's dropped off the products or dumped off the products, the enzyme is left unchanged. That is a key principle that you need to know. You need to know the enzyme is unaffected by the process. So what factors influence enzyme activity? One is concentration, two is pH, and then the third one is temperature, and we're going to look at each of these individually. So concentration is two ways. One, you increase the concentration of the substrate, or two, you increase the concentration of the enzyme. So this is a graph showing you how basically this works. You can increase the enzyme concentration or the substrate concentration, and the activity is going to increase until it starts to level off. What we call that, this area right here where it starts to level off, is the saturation point, meaning you're not going to get any more activity out of it because it's not physically capable to do any more work. It's as if, if I give you 10 worksheets and you're only capable of doing 5 worksheets in an hour. Great, I gave you 10, but all I'm going to get in an hour is 5 worksheets. So increasing concentration, the activity is going to increase until you reach that saturation point. Once saturation point is reached, the activity is going to level off because you're not going to get anything beyond basically that optimum range for the enzyme. pH is basically there's an optimal range at which an enzyme works, and usually you'll see that the activity is going to increase up until the optimum range, and then if it's exposed to temperatures or pH beyond that optimal range, then you'll notice that the activity decreases. And the reason that is because the enzyme has become what we call denatured, and you'll see the spelling of that on the next slide. And basically what that means is the enzyme has lost its shape and therefore its function, because remember, enzymes are reliant upon its shape. So just like a water bottle, if you take a water bottle and you put it on a stove and you heat it up, it's going to melt. If you then try to pour water back into it, chances are it's not going to be able to hold water because it's melted it severely. And that's exactly what happens with enzymes. If they're exposed to temperatures or pHs beyond their optimal range, they become denatured and therefore they lose their function. So temperatures, same way. They're going to increase up until the optimal range, and then once they're exposed to temperatures beyond their optimal range, notice that you have a decline in function, and that's because the enzyme has become denatured. It has stopped basically being able to work because it's lost its shape. And for temperature, as the temperature increases, activity is going to increase up until an optimal range. It's just like if I asked you guys to go outside and shovel snow in a very cold temperature. You can go out and do it, but you're not as active because it's temperatures are too cold for you to work. But as the temperature increases outside, then your basically your rate of action would increase because it's getting warmer and warmer up until the point where you get too hot, and then it's difficult for you to work. And that's exactly how enzymes work. They're going to basically increase activity up until their optimal range, and then if they're exposed to temperatures beyond their optimal range, they're going to stop work. So you need to know the factors that influence enzyme activity, and that is concentration again, temperature, and pH. And you're going to get to see how each of these work in some hands-on activities that we're going to do in class. You'll see, for example, catalase. Catalase, you heard me talk about that in class earlier. And that is an enzyme that is catabolic. It's going to break down hydrogen peroxide, because hydrogen peroxide is bad for us if it builds up in our cells. So all living things have the enzyme catalase because it's going to break down hydrogen peroxide into the two basically non-harmful components, which is oxygen gas and water. So if you ever poured it on your wound, you've seen it bubble up because it's producing oxygen gas, because it's breaking down hydrogen peroxide to create oxygen gas, which is O2, and then water, H2O. And you'll see that reaction in lab later on in the week.